Our lab is my molecular genetics. I'm Jaelynn Canty.
Aolani Valderrama. And I'm Emerie Obregon. In our agenda, we have the introduction, some background, and this lab has four procedures. This project will show how some molecular biology techniques work, such as PCR, VNTR, RFLP, and microarray analysis. Our hypothesis is if molecular biology techniques are applied to analyze DNA, then unique genetic patterns can be identified because um, these methods target individual specific variations.
Okay. So something to help out with this lab is a little background on cells and the information that they provide. DNA structure actually tells the cells its activity. We learned of this through central dogma, which is genetic information from replica DNA, which is then transcripted into RNA, then translated into protein. This is also shown in gene expression.
Okay, and then a little about procedure one. The PCR amplification of DNA is taking a specific sequence of DNA and it's making copies for this lab. You'll need to be familiar with using micropipettes as well as being able to measure the distance of a marker in the band. We also have to remember one band is homozygous and two bands is heterozygous. And then for procedure two, we'll have to analyze a DNA fingerprint by using VNTR, which is variable numbers of tremor repeats. And then we will also have to calculate the repeats in each alley, and um see the genotype frequency with looking at for the, sorry, I don't know how to say it, homozygote or heterozygote.
DNA fingerprint can be found using restrictive fragment length polymorphism, or RFLP for short. RFLP is a process of a unique pattern of DNA fragment created from when an enzyme is used to cut a DNA. Researchers use this technique to help with medical research as well as forensic investigation, and as you will do for this lab, a paternity test. In this lab, you will be given three mother DNA and three father DNAs, and you will use them to determine who are the parents of the child DNA that is provided. Use this practice to understand um, how DNA from a mother and a father can be used and understand how you figured this out.
On procedure four, um, we're gonna be looking at microarray analysis. In this procedure, we're gonna be seeing samples
from skin cancer cells and from healthy cells from a patient's um, skin. Um, the we're gonna be using mRNA from the patient's cancer, which is gonna be labeled with red fluorescent marker, and the healthy cells they're gonna be marked with a green one. And it's gonna show in this little like table the microarray. Then students will have to analyze which genes are the ones with that are high in cancer or high in cancer cells or high in normal cells. Then with the color key provider, they're gonna identify like good and safe therapy and medication options for these patients. Also, adding up to this procedure, they're gonna be comparing the microarray from a father and a daughter, and they're gonna identify similar genes that they share, either if that gene has cancer or they're normal or equal in both. After this lab, students should be should be fa should be familiar with PCR react as the PCR technique, which is amplifying certain sections of the DNA. Also, they should know and recognize um, the BNTR and RFLP analysis. Also, they should know how to read in microarray analysis.